Recording.
Excellent. So good afternoon, everyone, and welcome to this webinar in collaboration with Domainex. It's actually the fourth webinar in a really interesting, very informative series that we've held in collaboration with Domainex on their technologies. And if you'd like to catch up on the previous episodes, then you can indeed find those recordings on their website. But the content for today's webinar is on the topic of the reemergence of covalent compounds, Domainex's approach to covalent fragment screening. So there's been a rise in the use of covalent inhibitors in drug discovery, which has partly been fueled by technological advancements. In today's webinar, Domainex is going to cover one aspect by talking about their approach to covalent fragment screening. So I look forward to hearing from Domainex's experts about what we can expect from these approaches and how it will go forward to support innovation. Before I hand over to the expert speakers today, just a little bit of housekeeping from myself. So if you're not speaking, then it would be greatly appreciated if you can remain muted with cameras off. You'll have heard that we are recording the session. So what that means is that afterwards you can revisit and recap on the lessons from the content today. And then indeed, if you feel like there's somebody who would have benefited who you know is not online today, then you can share that recording with them. And then finally, just to let you know that the chat box is enabled. So if you go to the bottom of your screen and you select chat, then you can use that to submit your questions, thoughts, comments. And after the presentation, I will pick those up and sort of feed those to the speakers. So before we crack on with the webinar today, I'm going to play a short introduction on Domainex that has been put together by Natalie Winfield, who is Executive VP of Commercial Operations at Domainex. So if you just bear with me, I'm going to play this short video first. Thank you.
Hello, thank you all for joining this webinar. I'm Natalie Winfield, Executive Vice President of Commercial Operations at Domainex. I'm going to give a very quick introduction to Domainex before we get into the science. So for those of you who don't know much about Domainex, we're a fully integrated provider of preclinical drug discovery services. We're based in the Cambridge, UK area and currently have two sites. Our site at Chesford Research Park houses our chemistry and bioanalytical departments, and we opened a new site at Unity Campus for our biology department at the beginning of 2024. We have received multiple awards for our services, including most recently the prestigious King's Award for Enterprise. We have a world-class team and currently have over 100 staff. Our scientists are highly qualified and experienced, with around 90% having either a master's degree or PhD. They have a wide range of expertise across disease areas and target classes. We have a global client base and work with a range of different types of companies, including biotech and pharmaceutical companies, academic institutions, and patient foundations. We offer the full range of preclinical drug discovery services, including protein production, assay biology, medicinal, computational, and analytical chemistry. We support our clients through all the stages of drug discovery, from target validation and protein production through to lead optimization. If you would like to find out more, please do get in touch. I hope you enjoy the webinar. Thanks.
Okay, brilliant. So with that, I'm now going to hand over to Andrew Ratcliffe, Director of Chemistry, and Scott Martin, Head of the Bioanalytical Science Department at Domainex, to take us through today's presentation. So over to you both. Thank you very much.
Great, thanks for the introduction, and it's a great pleasure to be with you today. I'm going to start the presentation talking a little bit about the history of covalent drug discovery, and a little bit about our covalent fragment library, and I'll pass on to my colleague Scott Martin, who will cover some of the bioanalytics of fragment screening and some of the follow-on studies one can do. So a little bit about why covalent drug discovery. What's the advantages of covalent, working in covalent drug space? So one potential advantage is an increased duration of action. And that's very much dependent on the protein of interest resynthesis TR. So if you take, for example, BTK,
Oh,
the estimated...
Sorry to interrupt. I can't see the slides. Can
Okay.
have maybe you could reshare them. I'm so sorry.
Sorry.
I'll reshare them.
Sorry.
Let me do it.
Go back.
Mhm.
Okay.
Perfect.
Is that okay now?
Yep, perfect.
Apologies
Thank you very
for
much. Yeah.
Apologies for that technical hiccup there.
Apologies. Thank you. Yeah.
I'll start again. So
Thank you.
that's a pleasure. So just thinking about the potential advantages of working in covalent drug space. One of them is potentially an increased duration of action. And that's very much dependent on the protein of interest resynthesis TR. So if you take, for example, something like BTK, the estimated protein resynthesis in human B cells is about 48 to 72 hours. So consequently, knocking out BTK should give you a relatively long runway in terms of biological inactivity, probably way beyond any plasma levels of BTK inhibitor remaining. That should lead to less frequent drug dosing and should limit the likelihood and severity of drug-drug interactions and adverse events due to off-target activities. I also highlight here reduced pharmacokinetic sensitivity. And what I mean by that is that often with reversible drugs, you are optimizing PK to give you a certain plasma level at an extended time point. So for example, 12 hours or 24 hours. That's not necessarily the case with covalent drugs, where it could be very much a hit and run situation whereby you hit the target hard and you get a long duration of action, but the compound's already left the circulation. There's a very good track record for covalent drugs in successfully targeting intractable shallow binding pockets and discovering cryptic pockets that can be subsequently exploited. And then finally, as of today, pretty much now covalent drug discovery is disease agnostic. Looking at the history of covalent drugs, then they go back probably to about 1899. In fact, most drugs pre-2000 were from marketed drugs where the mechanism of action was later discovered to be covalent in nature. So again, starting with aspirin, 1899, this was shown to acylate a serine in the COX-2 enzyme. 1928 saw the launch of penicillin, and in this case, you get a lactam opening bicatalytic serine of a penicillin binding protein. And then 1988, you saw the launch of a protein pump inhibitor called omeprazole, and that forms a disulfide bond with a cysteine in an ATPase. And then 1997, you saw another protein pump inhibitor marketed called chlorapidogrel. And here you get a metabolic activation of the thiophene to an active thiol, and that forms a disulfide bond with a cysteine in a P2Y12 purogenic receptor. I've actually termed 2000 to 2020 as a sort of an era where there's a rise of the designed oncology covalent drugs. And the drive behind oncology as a therapeutic area was the perceived safety risk of covalently modifying proteins that could cause autoimmune reactions such as idiosyncratic liver tox in non-oncology indications. This has been a very rich era in terms of certainly the kinase area. So starting with ibrutinib, 2013, that's a BTK kinase inhibitor. About the same time, you've got afatinib, which is an EGFR kinase inhibitor. Now, one of the challenges with afatinib is it's got dose-limiting tox in terms of GI and skin. However, when you look at osimertinib, launched in 2015, this targets an EGFR kinase gatekeeper mutant, but more recently, it also has selectivity against the wild-type EGFR kinase, which causes the GI and skin toxicology. So consequently, it's got a much wider therapeutic range. And then finally, 2020, you've got sorsorotib. This is a kinase, KRAS kinase G12C mutant inhibitor. And here's a nice example of covalent drug discovery interacting with what's perceived a undruggable target at the time. And again, it really exploits the discovery of a cryptic pocket.
Scott, sorry, I'm just going to, can everyone see the slides changing? I just want to make sure that that's going through okay.
Yeah, they seem fine.
Are they? Yep, all good. Thank you. Thanks.
Great.
Okay.
Thanks very much.
You. Yeah.
Recently, more recently, there's been a mind shift to non-oncology covalent drugs. So here's some exemplars. Looking at the top left, this is actually derived from a natural product. It inhibits KEAP1. It's approved in the US for Friedreich's ataxia, and Biogen acquired this compound for $7.3 billion. And then the bottom left is another BTK kinase inhibitor. This one's designed for the CNS. It's undergoing regulatory submission for MS, and Sanofi acquired that for $3.7 billion. So again, two exemplars which show you the power of or the return on investment of working in covalent drug discovery space. And then on the far right are two nice examples from Pfizer. The top right is a JAK3 selective kinase covalent drug approved for alopecia. And again, alopecia is not a life-threatening disease, but again, I think that highlights the potential safety associated with covalent drugs. And the bottom right is a nirmatrelvir, it's a COVID-19 drug, and Pfizer have dosed over 50 million patients that worldwide. Again, I think that just, again, talks to the potential safety of that particular drug. There are two essential approaches to discovering covalent drugs. The first one is reversible first. This relies on identification of a potent reversible structure where a covalent warhead can be attached that interacts with a nucleophilic target residue. And this plays very much to the strengths of structural based techniques. A nice example is osimertinib, where AstraZeneca profiled some reversible EGFR kinase inhibitors, discovered compound 1, and then used that as a scaffold to develop osimertinib. The second approach is called electrophile first. This relies on stringing a library of fragments containing a reactive handle, and again plays to the strengths of structural based techniques, but also to mass spec. A nice example with sorosorosib is the fragment 2, which is an acrylamide zetadine that was used as the initial fragment that moved the program forward. Also, interestingly, is the possibility to develop reversible covalent drugs. And here this offers a further strategy to mitigate potential off-target activities by irreversible binding to non-target amino acids. And here the challenge is focusing on tuning inhibitor off-rate residence times. Now, there are some exemplars. Bottom left is another BTK kinase inhibitor that's in phase 3 for immune thrombocythemia. And in fact, the reversible covalent warhead is a cyanoacrylamide. Then moving to nirmatrelvir, of course, that's a cyano reversible covalent warhead. And then two further classes worth highlighting is the cyanamide class. This is a privileged scaffold for DUBs. And then the dichloroacetamide class on the far right. It's a great strategy for reducing the intrinsic warhead reactivity of chloroacetamides. Most marketed design covalent drugs target cysteine. However, over the last few years, there's been a great interest in developing covalent inhibitors against other amino acids. The plot on the left is a number of publications against over time. You can see that there's a great interest over the last few years in tyrosine and lysine and threonine. And on the far right, a table giving the amino acid percent prevalence in human proteome. I've highlighted cysteine at 2.3%. You can see, for example, that lysine and threonine are much higher percent, around about between 5 and 6%. Some non-cysteine covalent warheads that are commonly used. You've got the fluorosulfonate class and the sulfonyl fluoride class. They've been used for targeting histidine, tyrosine, and lysines. And then the bottom, you've got the aromatic aldehyde class, which targets lysine. And then finally, a masterclass in covalent drug design from Revolution Medicines, just recently published this year, RMC 9805. This targets a KRAS G12D, so we're actually targeting an actual aspartic acid mutation that's found in pancreatic and colorectal cancer. And what's fascinating about this particular compound is, in fact, the covalent warhead is an aziridine. It also has a unique mechanism of action whereby 9805 first binds to cyclosilin A, and then the binary complex then binds to KRAS G12D before triggering a covalent tethering mechanism. And this compound's completed phase 1b orally administered. And given the fact this compound sits in the beyond rule of 5, against a testament, I think, to the great drug design from Revolutions Medicines. They've had encouraging safety signals and preliminary efficacy readouts from that phase 1b study. Just moving to the Domainex covalent fragment library design. What we did here was we took 16 targeted covalent fragment offerings from 5 suppliers. We combined them. We then filtered for preferred warheads, and I'll describe those warheads in the next few slides. We then did some in silico property prediction and filtering for fragment light space. And then looking at clusters of warhead, we then used Schrödinger tools to look at diversity of the non-covalent fragment space, and then used a visually chemically sorted 2D map to choose fragments in that diversity space. We ensured the fragments were tractable, and then we actually brought in 690 fragments overall. This is the profile of the library. It very much sits with the rule of 3 in terms of molecular weight, log P, H-bond donors, and H-bond acceptors. And the actual warhead profile. You can see here the pie chart on the top right and the table below. We actually added 35 synthesized cyanamides to this collection. It's very hard to get hold of commercially cyanamides of real diversity. So the library contains both reversible and irreversible warheads, the reversible ones being the cyanamides and the cyanoacrylamides. By far, the highest percent covalent warhead is the acrylamide, and really that's because it's a prevalence in marketed compounds and in commercial compound collections. Should also mention the utilization of high-throughput chemistry direct to biology. This is screening of unpurified mixture to rapidly identify covalent inhibitors worthy of further investment, or, for example, rapid SAR exploration of fragment screening hits. 2 really nice examples here, one on the left from GSK. Here they took a library of amines and formed a library of sulfonyl fluorides, screened that library unpurified using mass spec techniques, identified hits against carbonic anhydrase 2 and BCL6, and then followed those hits up with further investigation. On the far right is a nice example from Near London's group. Here they took this acrylamide alkyne and then using commercially available azides formed a triazole library. They then screened that unpurified in a high throughput in-cell Western assay, identified potent inhibitors, and then resynthesized a singletons purified compound, and then ran some further investigation of those compounds in terms of LCMS and crystallization. We've invested heavily in the HTC D2B at Domainex, both in the non-covalent space, but also covalent space. Here's an example of a workflow that we've performed. We mined our internal registry for amines less than molecular weight 300. We then removed reactive species and multiple amines and paints, did a little bit of in silico property prediction and filtering for fragment-like space, and then chose basically 96 fragments from some Schrödinger tools. And then we essentially reacted those 96 amines with 3 validated warheads to give 288 covalent fragments in less than 24 hours. Those 3 covalent warheads being acrylamides, chloracetamides, and propanol amides. And the way we utilize HTC D2B covalent at Domainex is we do a compatibility assessment with each protein first, using a known positive control available or iodoacetamide before we invest in the actual HTC D2B reaction optimization and library synthesis. I now pass over to my colleague Scott to talk a bit more about some of the bioanalytics and some of the follow-on studies.
Thank you, AJ, for that great introduction to covalent inhibitors. Today I'm going to talk about our covalent fragment mass spectrometry-based screening platform here at Domainex. So the initial screen is essentially an incubation of covalent compounds or fragments against a target protein, and then we measure the percent bound. We run the protein denatured, so we only measure covalent interactions, and we determine the stoichiometry and the quantity of binding. We can run this as single compounds or in pools of 5, and I'll talk about this in more detail over the next few slides. Once we get those hits from that initial screening, we then assess their intrinsic reactivity with a glutathione reactivity assay. And this is essentially an incubation of those compounds with glutathione, and then we measure their half-life and the formation of the GSH adduct. This is done real-time kinetics, so it's high-quality data. Typically, if the half-life is less than 100 minutes, we would think that those compounds are not really suitable to progress any further. So it's another way to triage compounds down to improve the quality of hits. Once we've reduced that number, we look at binding site ID, because it's really important to make sure that your covalent warheads are binding to the right cysteine. And it's even if you're using proteins where there is only one cysteine, we have seen off cysteine binding before. So it is always important to confirm that. We do this by proteolytic digest, high resolution accurate mass analysis, and then peptide mapping software to easily identify the binding site. Once we've done this, it's just to benchmark your hits with K-IK-NACT, and this is essentially a measure of the binding rate. And I'll talk a little bit about how we do this, and this can be taken forward to further optimize compounds. This is a flow diagram of our covalent binding MS screening workflow for that initial screening. So it is really important before moving to any screening of compounds that there is an assessment of the suitability of the protein for the mass spec screening. So the first thing to do is assess the mass spec response, then to look at how the protein behaves under certain incubation conditions, and we also assess with a couple of reactive fragments, IAA and MPA. Once it is deemed that the protein is suitable and we've optimized those incubation conditions, you move forward to incubating the compounds, and we do this in 2 3-8-4 well plates. So that means the capacity is about 750 compounds per overnight run. And the output from this screen is, in essence, a mass spectrum. On the left, you have unbound protein, and on the right here, you have bound protein. If there were additional binding hits, then you would see additional hits in the spectrum here. And then the output from this spectrum is that we actually use auto-processing, and we measure the level of binding for expected, unexpected, and the degree of multiple binding or stoichiometry. I will go into more detail about this whole process. Just to point out, there are occasions where the protein isn't suitable for screening, so there may be alternative approaches that can be used there, such as a mutated or truncated protein. So this is a typical example of a feasibility assessment. So as I said, first things first, you do a protein QC to determine the response, but also to make sure that that protein is pure. It'd be very difficult to screen if it was an impure protein, because the mass spec signals would interfere. There is also a standard incubation assessment that we do, which is an array experiment of 3 concentrations, 2 different incubation times with 2 different temperatures. We also assess a quench reagent and check the stability after 24 hours and sometimes 48 hours. So it's really important to make sure that your protein is stable over this incubation period, and the output from this feasibility assessment is shown on the right in these graphs. If we just take a look at the 37°C IAA results here, the red is protein no binding, the blue is single binding, the purple is 2 binding events, and the black is 3 binding events. This is real data from an assessment that we've had in the past, and we selected the most optimized conditions to go forward into the screen. It is really important to do this, as I've said, this re-injection at 24 and 48 hours to absolutely confirm that the quench reagent has worked, because these runs of up to 750 compounds are often about 16, 17 hours. So you're happy that the protein is suitable for mass spec screening, it's stable, and you're able to get some reasonable results with those incubation conditions. So you move forward to the MS screening. Typically, as I said, we would run 2, 3, 8, 4 plates. This would generate 750 data files to process. And I'll just take you through how you would process each data file manually initially, and then I'll talk a little bit about how we do it automated. So the first step is to generate your charge envelope spectrum from the chromatogram. Once you've generated that, you process that to a much simpler spectrum with just the masses of interest. This is a spectrum from the IAA experiment, so you would be looking for the expected addition of IAA. And lo and behold, we have what is 3 additions of IAA. Now that is the expected mass additions. To check for unexpected mass additions, we would insist on running a negative control, so you can compare a negative control spectrum, and you can simply eyeball both of these to see if there's anything in that control sample that could interfere with the process spectrum, but also if there's anything additional in the process spectrum that isn't in the control, that could be another binding event that may have been missed. Manually processing can take up to 5 minutes per sample, depending on how complicated they are. So 750, this is 60 hours of processing, which is weeks of processing time. And you really wouldn't want to be staring at spectrum day in, day out. So what do we do to process this data? So to process this data, we use our automated software, and I'll describe how this works. So on the left, you have a graph with expected binding hits, and the expected binding hits are determined in the software by measuring that expected mass addition to within plus or minus 10 ppm, which for a typical protein between up to 100 kilodaltons would be 1 to 2 daltons. We also look at what we refer to as spectrum change, which is the unexpected hits, and spectrum change measures the difference between that control and with protein with no binder and the sample. And you just see here there are a number of hits in both of these. Now this dotted line here is a threshold, and that really is determined by the signal to noise in the spectrum, and you can see there is quite a bit of noise here. We would advise that you would maybe use this threshold to optimize the hits, and if you had an awful lot of hits, you would probably raise that threshold up. If you had fewer hits, you drop that threshold a little bit, but that can be done post-processing. So that is the two of the things that we measure, and you can simply then plot the expected binding versus the spectrum change on a graph like this. And those which line up correlate quite nicely are all the expected binders. Then these where there is a measurement in the spectrum change, but no measurement or no significant measurement in the expected binding, we will determine these to be unexpected binders. Now this can be due to a different mechanism. It could also be due to an in-source liability. And these things can happen inside of the mass spec source, and you lose water or ammonia. Whilst the actual hit is valid, the mass software won't pick up these -17 or minus water that could happen in source. So it's good to have this spectral change or spectrum change assessment, and this will highlight these unexpected hits. Now we have seen these hits in the past due to different binding mechanisms, due to these in-source liabilities, and also in some occasions artifacts. Then there's a third option here, which is expected and unexpected binding, and this we've seen recently where we had a loss of water, and that was half of the binding was the intact molecular ion, and then the other half was due to loss of water. So in reality, you would just sum those and actually put those back up in here. But it highlights that you could underestimate the amount of binding if you don't quantify all of the ions relating to that binding event. And I'll move on to how we determine stoichiometry next. So how do we calculate the stoichiometry? Well, looking at the example on the left, you have protein and 3 binding events. Now, how we determine the stoichiometry using software is that the software will sum all of the expected binding events above the first binding event. So binding event 2, binding event 3. Now that sums the peak intensity. So that is a measure of how big the binding event is, but also how many binding events. And you sum all of them, and then you will sum all of the binding events, including the first binding event, and simply divide one by the other. This enables you to generate a ratio, as we've done with the binding intensity. And the closer it is to 1, the more multiple binding events there are, and the closer it is to 0, the likelihood is that there is a single binding event. And what you can do now you've got this stoichiometry data as a ratio is you can plot that against the binding quantity. And you can see here we've got this graph, and what we have done is we've arbitrarily applied 4 quadrants, and this just helps you to interpret the data. So on the bottom left, you have weak to low, weak or medium binding activity with a stoichiometry of 1 to 1, and you can see that in the spectrum here. It's just a single binding event, and it's a small binding event because the ratio between the protein and the bound protein is quite low. And then you can move to the high binding activity event in the bottom right hand corner, which is probably more where you'd want to be from these hits. And you can see here single binding event, and the protein bound peak is much bigger. And then the third quadrant, the top right, you have got high binding, but also you've got multiple binding events here. So you can look at this data very quickly and identify which hits are probably your highest priority, and that would most likely be either those on the bottom right or maybe some of the weaker ones that have single binding events. It's just a really nice way to process and view the data quickly. So another way of representing the data is to go back to the library, and this is our in-house library that AJ described earlier. And what we can do is we can look at the quadrant here, the highly reactive and multiple binding events, and see which of those warheads are generating all of those hits. And you can see here from all of those different warheads, they are all chlorocetamides, which are probably very reactive, which explains the off-target binding. So it's just another way to view that data, which is quite nice. So to summarize how we use this data to identify hits and then triage them down to optimized hits for further assessment, we first off filter on unexpected binding events. So we have that threshold at 0.1. Anything above that is seen as a hit, which gives you these 81 hits here. And this is a real example. You then filter on the binding stoichiometry, and anything above 0.01 is removed. So anything with any hint of additional second or third tertiary binding is removed, and that reduces the hits down to 45. And then at this point, we can assess the spectrum change versus expected binding. And if you see on that earlier plot, there were 4 hits that were not expected. So at this point, it's worth investigating those further hits. And if they're artifacts, you just take forward the 44, 41. But if they are due to in-source liabilities or there are other binding events, it might be worth investigating these as well. And that's what we end up with, 41 hits moving forward into the GSH reactivity assay. This is our in-house glutathione reactivity assay, and glutathione is a simple tripeptide here with a cysteine, glycine and glutamine. It is also prevalent in the body and is a detoxification mechanism for reactive electrophilic metabolites or compounds. So the way that we do this assessment is to simply incubate the compounds with glutathione and measure their depletion over time and generate a half-life, but also we measure the glutathione adduct formation. And this is all real-time kinetics. We do not use quench reagents at all at this point, so the data is of high quality. We also use a PBS control, so we check a compound that is spiked into PBS, phosphate buffer at the start, and then we check that sample at the end of the run, and we just make sure that they are within a reasonable percentage of each other to ensure that any of the compound depletion is not due to the stability of that compound. We are able to run this as a single compound analysis or in pools of 5. The advantage of running them as a single compound analysis is that we are able to acquire UV data, which is a more accurate way to measure both the formation and disappearance of glutathione adducts, and is more of a normalized response, and I'll show that in a little bit. The advantage of running 5 compounds in a pool is obvious. We tend to apply a 5 Dalton filter. The reason we apply a 5 Dalton filter is because we want to be sure that there are no mass clashes in those pools, and it is only a mass spec readout, so it's not possible to generate UV with these mixtures. And again, we would make it very clear that compounds with less than 100 minutes are probably not worth progressing. Those with between 100 and 200 minutes are worth flagging as maybe on the high end, but over 200 minutes, we believe that's probably a good compound to take forward. So just to show you some of the data that we generate here. So this is nitrophenol acrylamide, and it's measuring the turnover and the formation of its adduct on the left by mass spec. And you can see the MS response here of the compound is very much lower than the MS response of the glutathione adduct. And you get the glutathione adduct achieving a higher concentration or a higher response than the initial compound. So you don't really get a very good mass balance here, and you can see the data is a bit more up and down. If you have a look at the UV response here, you can see that the response is more normalized and that the data is much smoother and much higher quality. This enables us to make sure that we're getting a mass balance with the formation of the glutathione adduct, but also the disappearance of the test compound. As another control, similar to the PBS control, to make sure that something else isn't happening here that we're missing. And then we just generate a half-life, and it's 26.4 minutes in our assay for nitrophenol acrylamide. So what about compounds that are reversible? So if you have a look at the graph on the bottom right, on the bottom left here, you can see there's some mass spec data for test compound and glutathione adduct. And this is measuring, again, compound disappearance and glutathione addition. But you can see that GSH is present from the very first time point, and test compound doesn't really drop off over time. In fact, it's a plateau, and it looks like they're in equilibrium. Now, this would suggest that there's reversibility here, and we have seen this with a number of compounds in the past, and typically, by the first time point, they reach an equilibrium. We have seen some compounds that take a little bit longer to reach that equilibrium point, but normally it's a very early time point, if not the first time point. So this is demonstrates that this is a reversible test compound. Now, because it's mass spec data, it's not possible to generate a ratio of the glutathione adduct versus test compound, and I'll show why that is in a second. Because when you look at the UV data, you can actually generate a ratio between test compound and GSH adduct, and this UV data is from the same compound that's shown on the left. So the GSH adduct, as I mentioned previously, has a very different response by mass spec than the test compound, and this is typical of glutathione adducts. But what you can do by UV is you can measure the amount of test compound, measure the amount of GSH adduct, and generate a percentage, because this is more of a normalized response. And actually, you can take each time point and ratio those, and actually it's 4% across all of the time points. So it's just some more information you can obtain by UV around where that equilibrium point is. It is also important to note that there is advantages of running reversibles, and you get less off-target binding, which is much more helpful for safety, etc. So once you've triaged your compounds down through optimizing on the screen and through the intrinsic reactivity with GSH, you then end up with a number of compounds more amenable to looking at the binding site ID. And that is really important to make sure that you're hitting the correct targeted cysteine. So how we would do it here is we would incubate the compound with the protein, the target protein, and we would potentially incubate that for a bit longer to make sure we get as much binding as possible. We would then use protease digestion to break up that protein at specific points that are known, and then you end up with a soup of peptides, and one or two of those peptides may contain or should contain that covalent compound. How do we determine which peptide or where that peptide is, that compound is bound? We run high resolution accurate mass, and we generate a nice chromatogram with lots and lots of peptides, and then there's peptide mapping software which we use to then identify the binding site. So it's a way of deconvoluting complex data. But I just want to give a quick example of how we would do this. So before we would start this experiment, it's key to look at your protease enzyme or the method you're using, and for us it's trypsin as a standard method. And trypsin cleaves at lysine and arginine, and you can see with the protein sequence that I have under here, I've highlighted all those places that you will get cleavage. So if you were to be targeting this cysteine here, then that is quite a good size peptide, and I think you could just move on to the actual experiment, and you should be quite easily be able to identify that as the binding site. However, if it was one of these other cysteines up here that you were trying to target, then that would be much more difficult, mostly because the peptide is very much bigger, but also there are two cysteines in that large peptide. So it might be worth considering an alternative proteolytic enzyme at this point. It is also worth pointing out that even with the previous example where it looked like you had a nice size peptide, that you can get miscleavages, and the standard methodology might not be suitable to generate a nice peptide map. So as I mentioned, the method might not be suitable for identifying the binding site. So we have a number of ways to optimize this. Firstly, if you have issues with maybe the protein not being completely denatured for the digest, we can look at different concentrations of urea or different denaturants such as guanidine instead of urea to try and unfold that protein and get a better reduced miscleavages and a better coverage. There's also alternative digest enzymes that we can look at using, and I've just on the right hand side here you can see there's a bit of an example where we use different digest enzymes or proteolytic enzymes to break up a protein sequence here. We can also look at assessing different reducing agents and other things. Just to move on to the next slide. So I just really wanted to show on this slide how many proteolytic enzymes there are that we can use to improve that digest methodology. I'm not going to go into this in any detail. It's more just to give you an idea of the options that we have. Once you've completed the peptide mapping, you end up with a result that looks like this, and you can highlight the binding site quite easily in the software. Now, we also generate MSMS data here, or MSE data, and that enables us to get a fingerprint of that peptide and pin it down exactly on that cysteine. If there were more than one cysteine in that peptide, if the peptide wasn't too big, we should be able to deconvolute that mass spectra and pin it down to the exact cysteine. So once you've optimized your hits, what we like to do is benchmark them, and we use KNet KI to do that. This is simply a measurement of the rate of covalent binding of the compound to the protein, and it's assumed that that covalent binding inactivates the protein, so it can be used as a measure of potency. The first step in this binding is the affinity and selectivity. So this is the non-covalent binding event here, so you get your compound in the pocket, and then the K-inact is where the covalent bond is formed with the cysteine. Now, the K-inact can be due to the warhead reactivity, but you also have to remember it could be due to the orientation or distance of the warhead to the cysteine, so it's not purely reactivity. And then it also could be due to the reactivity of the cysteine itself and the surrounding amino acids and the pKa. But with the cysteine reactivity, it should be the same across all compounds. So I just very, very quickly want to explain how we generate the KNet KI data. And KNet KI requires you to incubate your compounds with protein at different concentrations, but also at each of those concentrations take multiple time points. So it's quite an extensive piece of work. What we would do before we embark on this KNet KI experiment is to look at a single concentration, which is normally the concentration that we screened at, and measure up to 10 hours time point. This is really important because what you need is your compounds to be within the range of the experiment. So you need to have the right concentration range and the right time course. And we have a real example here in the bottom where we have generated formation of the bound protein over time, and you can see it over the time course, and this is run at 50 micromolars. We refer to this as the K observed. And how do you calculate the KNet KI? Well, you then redo a number of concentrations and time points based on the data you got from the initial K-obs experiment, and you end up with a number of K-ob values, and that is the slope of each of these curves. And all you need to do then is plot the slope of each of those curves against the concentration, and that enables you to generate the KNet KI data. I know this is only a brief look, but that's all I was going to talk about in this presentation. Thank you. I would like to acknowledge a couple of people from the bioanalytical department, Gary Wu and Jason Pembroke, who pulled together a lot of the data for this presentation and are actually online in this webinar if there are any particular technical questions around anything that I've presented. And then for chemistry, Anna Hopkins, Graham Sloan and Fergus Preston to help curate the Domainex library. Thank you. And now we'll open up to questions.
Excellent. That was very, very informative indeed. Thank you so much, Andrew and Scott. So the chat box has been enabled. So now is your opportunity to speak to the experts directly. If you'd like to answer any questions or any comments on what you have just seen. If it's easier for you to raise your hand using the reactions tool at the bottom of the screen, and then just speak, please do feel free to use that instead, and I'll keep an eye on the list of participants. Excellent. So while we wait for any questions, oh, I have one there. What is the throughput of the GSH reactivity assay?
So I can answer that. So it depends if you run singles or if you run pools. If you run singles, we can run 10 compounds in a single run, and we can run 5 a week. And equally, if we do it in pools of 5, that's 49 compounds in the control. And again, we can run 1 every night. So up to probably about around 250 a week for pooled and around 50 a week for single.
Thank you. Do you have a standard fragment library as well as a covalent one?
Yes, we do. We have a standard fragment library of about 1,200 fragments, which again has been curated. And in fact, we have a very good track record actually of that library generating hits across multiple targets over the last couple of years. It's a very powerful library.
Okay. Thank you. And what is the usual molecular weight range for a protein that is feasible for mass spec screening? I hope I'm doing these acronyms right. And can you do screening via peptide mapping? For example, if a target protein doesn't work well in mass spec, are there a downside to this? Yeah, so firstly, with our experience, less than 100 kilodaltons is a good start. I think around 50, 60 is probably where it starts to go a little bit more difficult. But you have to remember, you don't have to run the full protein. You can screen a truncated protein as well, and we've done that in the past. That's worked quite well. And in answer to the question about the digest, yes, you could do it by digest, but I don't think the throughput would be anywhere near as high as it would be with the intact mass experiment. So there are advantages to running the intact mass, I think, and it's a little bit quicker and easier to process that data than it would be with peptide mapping. But yes, if it was difficult to get that intact mass, you could go down the digest route. Okay. Matthew, did you want to unmute to answer yours? Sorry. Oh.
Yeah, so I've got 2 questions. The first question is the glutathione assay where you excluded hits that had half-lives less than 100 minutes after you found them as hits. Why don't you just exclude them in the first place? Anything with a glutathione half-life less than 100 minutes. So that's the first question. And then the second question with the feasibility assessment, you only used iodoacetamide and an acrylamide. Does that mean that you're only assessing it for cysteine reactive electrophiles and not other electrophiles that are reactive with other amino acids? That was the 2 questions.
Great. Thank you. I'll answer the second one first. So that was just the example that we used as a typical feasibility assessment. So yeah, you're right. If that's a targeting cysteine, or if you're looking at a standard sort of acrylamide warhead, then that's something that we would do. It doesn't mean that that's a standard feasibility assessment. We can run other compounds, and we can run other reactive warheads to assess that binding feasibility. It's more around making sure that we understand that the protein's stable, and we can get a good signal, and we know what concentration we're screening at. But yes, we could definitely run different covalent warheads for if you're looking at different covalent warheads or targeting different residues other than cysteine. The other way that we've done this in the past is, instead of screening 750 compounds right from the start, you might want to screen 50 of a library, and we can assess on those 50 the conditions that we would then screen the wider library of maybe 1,000 or 2,000. So there's a couple of options there. In terms of the GSH, I think from our experience, I think osimertinib is a drug that has a half-life of about 120 minutes. And I know that is quite reactive. It's quite difficult to get ADME data and other things on that. But what you have to be careful of is glutathione is present throughout the body. So if it is reacting very quickly with glutathione, you're unlikely to get the exposures you need in humans. Also, if it is very reactive, you're going to end up getting a lot of off-target binding to cysteine. So I think it's an arbitrary 100 minutes, but I think I think it's just a number that we use to try and triage the compounds down. If you had 3 hits and all of them were less than 100 minutes, then obviously you'd need to think about that.
But when you've
So my question was just why not exclude anything that's too reactive in the first place before you do the screen? That was my only question. So if you've got compounds in the library that are more let's say they've got a half-life of 20 minutes, why are they there in the first place?
It's a good question, Matt, and it's something that we're actually doing at the moment.
Can I just answer quickly? So yeah, in our library, that's something that we're planning on doing, and we will assess all of those. Sorry, I misunderstood the question. We will screen our internal library, but often we work with client libraries as well, and we don't know what the reactivity is. And the quickest way to generate the data is to do the protein screen. That's cheaper than running the GSA chassis, and the throughput's much higher. So I think you're absolutely right. With our internal library, the plan is to do all of the GSA chassis on all of those compounds, but often we screen client libraries and we don't know.
That's work in progress, Matt. So it's a really good question, and we're certainly working at that as we speak.
Yeah.
Thank you.
Excellent. Thank you, Matthew. Thank you. So one further question. I'm not sure if you hinted to this before. So approximately what quantity of protein is required for this screening, e.g. molecular weight, around 50 kilodaltons?
Yeah, so we would start with about 5 micromolar for the feasibility assessment, but I think we can go low as 1 micromolar or lower. But it very much depends on the response that you get from that feasibility assessment, which is why we'd always make sure we run that and determine what the minimum amount of protein we can screen with. I also mentioned that we can cocktail compounds instead of doing singles, and we've got data to show that there isn't a lot of difference between running cocktails and singles. There is some competition. But if you do 5 compounds, you can reduce the amount of protein you use by 5-fold. So typically it's around 1 or 2 micromolar, but we start the feasibility at 5.
Excellent, good stuff. And thank you everyone for all of your questions so far. I'm just making sure there's no hands up. No. Okay. And in terms of the techniques you've discussed today on the session, are there any others that you could kind of allude to that are sort of the next thing in terms of screening, or is this it? Or what's next, I guess is my question.
I guess I'll say our fragment library is very much designed towards cysteine. So I think there is a potential to put together a small fragment library that's non-cysteine directed, in particular histidine and lysine and threonine. So that's something that we're considering at the moment.
And we're also looking at purchasing an ASMS system, so we can look at non-covalent screening as well, an inline system in the future.
Excellent. Okay, so we're just up to the hour now. So if there's no further questions, if nothing, or if something does come to you post the session, then don't hesitate to reach out to One Nucleus if you're not already connected to the experts online today, or if you're already connected to them, then I'm sure they won't mind. We can certainly help with facilitating any follow-up questions if that does occur. So thank you everyone for joining us today. Thank you, Andrew and Scott, for your time on this session. And that actually brings us to the closing of the series of 4 webinars that we've held in collaboration with Domainex. So a lot of knowledge, and as with all the others, the recording will be disseminated in a follow-up email that you will receive from Domainex. So indeed, you can always use that to get back in touch if you'd like to discuss your projects further with Domainex. So I think with that, we will close the session. So thank you very much, everyone, and take care.
Thank you.
Thank you.
Bye-bye.
Thank you.
Thank
Take
you.
care. Bye.